duplicated gene that is evolving into junk DNA or simply a random sequence that resembles a gene. Sequencing entire genomes. The sequencing described above is only effective for fragments less than several hundred bases long. To sequence a plastid or mitochondrial genome, organelles are extracted from a cell. Their circles of DNA are isolated and then cloned and divided into several batches. The DNA is cut into fragments, each batch being cut with a different restriction endonuclease, and then each fragment in each batch is sequenced. In the first batch, we might have several hundred fragments, all perfectly sequenced, but we do not know the order of the fragments. The same is true of the second batch, but each fragment here will have some portion that matches one or several portions of fragments in the first batch. By aligning the equivalent regions in the two batches, we should be able to align all fragments in proper order. Sequences from the third batch act as a verification. To sequence the immense genome inside nuclei, each chromosome is isolated from the others and sequenced individually by the same method. Figure 15, 23 very long pieces of DNA are sequenced by cloning the DNA, dividing it into several identical batches, and then cleaving each batch with a separate restriction endonuclease. Each fragment is then sequenced. If we had only used endonuclease 1, we would have gotten fragments AF, but we would not know their proper order. By comparison with the fragments from a second endonuclease, we would see that the left end of fragments A, G, and L are the same. Fragment H matches the right end of A, all of B, and the left end of C, and fragment J matches parts of C and D, and so on. Genetic engineering of plants. Recombinant DNA techniques have made it possible for botanists to identify, isolate, and study the structure and activity of many genes. They also allow botanists to insert genes into plants that do not normally contain those genes. For example, cotton plants have received a gene from the bacterium Bacillus thuringiensis, which codes for a protein toxic to caterpillars but not to other insects or mammals. If the gene protects the plant, it could prevent as much as $100 million in crop losses annually. Botanists have transferred genes from desert petunias into normal petunias, resulting in plants that require 40% less water. If such drought resistance genes can be transferred to crop plants, the amount of water needed for irrigation will be greatly reduced. Tomatoes have been engineered such that the enzymes that cause them to become mushy are inhibited without inhibiting the other enzymes involved in developing flavor and aroma. The tomatoes can be allowed to ripen fully on the vine but are still firm enough to ship to market. These GMOs are not welcomed by all people. In many cases, it is argued that they are not being used to prevent starvation but rather to allow agribusiness to make more profit. The argument continues that rather than genetically altering tomatoes so that they can be harvested and shipped while green and hard, isn't it better to buy locally grown tomatoes, perhaps even ones that have been grown organically and harvested at the peak of their flavor? For example, golden rice has been genetically engineered to produce more beta-carotene, a precursor to vitamin A. Many people in Southeast Asia have a diet heavily dependent on rice, and with normal rice, many suffer from vitamin A deficiency. Golden rice could provide them with more vitamin A, which is good, but the counter-argument is that this is still a peasant diet, a poverty diet, and there is an alternative solution: provide people with more fresh vegetables, make them less dependent on rice. Rather than focus only on vitamin A, improve more aspects of the people's nutrition. Obtaining some genes is not very difficult, but at present, inserting them into nuclear DNA properly so that they can be transcribed and translated is not easy. A gene may code for an improved, more nutritious storage protein and should be expressed in cotyledons during seed formation, but if the gene inserts into a region of nuclear DNA that codes for root, stem, or wood characteristics, the engineered gene either may not be activated or may become active in the wrong place. Research on the nature of promoter sites and their interaction with chemical messengers is especially intensive. Many genes have been located that are expressed only in particular tissues such as cotyledons, wood, chlorenchyma, or epidermis. Their promoters are valuable because a cotyledon promoter can be attached to our foreign gene before insertion, increasing the likelihood the gene will respond to the appropriate chemical messenger, regardless of where it inserts into the DNA. Figure 15:24. The gene for a luminescent protein from fireflies was inserted into a plant cell in tissue culture, then the cell was induced to grow into a full plant. Apparently, this gene had a promoter that is normally activated in every cell because all cells are glowing. By attaching different promoters to this gene before the genetic engineering, it is possible to study when and where the promoter is normally active. Biologists hope to find promoters that, when attached to this gene, cause only specific cells or tissues to luminesce, then those promoters can be used whenever we want to specifically affect those types of cell or tissue. After a gene and promoter have been prepared, they are attached to an insertion vector, usually the TI plasmid from the bacterium Agrobacterium tumefaciens. The plasmid has been modified by botanists by adding genes for antibiotic resistance. The completed plasmid promoter gene is mixed with plant cells in tissue culture. They are allowed to grow and then are treated with an antibiotic. Those cells that did not take up the plasmid, and those that took it up but are not expressing it, are killed by the herbicide. Those cells that have taken up the plasmid and are transcribing the gene and translating the mRNA into protein are resistant to the herbicide and survive. They can be cultured further. The surviving cells are induced to form new plants, which in many cases not only carry the gene and express it but pass it on to their progeny when they undergo sexual reproduction. Viruses. In the past, much of the emphasis in studying viruses in plants was on preventing the spread of virus diseases in crops. Now many viruses are especially interesting as possible vectors for plant genetic engineering. Also, because most are RNA viruses, they contain many unusual enzymes that are useful for the experimental manipulation of nucleic acids. Virus structure. Viruses are extremely small particles that usually contain only protein and nucleic acid. They were originally discovered in 1892 as factors that cause disease, but were so small they could not be seen with a microscope and could pass easily through filters with pores fine enough to trap even bacteria. They were never actually seen until the development of the electron microscope. They were originally thought to be very small cells, but now we know that is not true. They have no protoplasm, no organelles, and no membranes. They cannot carry out their own metabolism independently, they must always parasitize cells of some sort. The great majority of viruses, especially those that attack plants, consist of only one or a few types of proteins and a small amount of nucleic acid. Plant viruses always have a simple morphology, either long or short rods or even round particles. Tobacco mosaic viruses are rods 15-300 nanometers, whereas citrus tristeza viruses are rods up to 2000 nanometers long. Spherical or polyhedral virus are common, often having a diameter of about 60 nanometers. The diversity of nucleic acids in plant viruses is great. Of the known viruses, the greatest number are retroviruses, which contain single-stranded RNA. Retroviruses are a source of reverse transcriptases needed to make cDNA. Many viruses that cause cancer in humans are animal retroviruses. 12 known plant viruses have double-stranded DNA, and there are 2 groups of unusual types, 10 contain double-stranded RNA, and 15 contain single-stranded DNA. Furthermore, many plant viruses are split genome viruses, not all of their nucleic acid is packaged as one particle. Instead, the virus consists of at least 2 different particles, and both must be transmitted to a new host cell for viral metabolism to occur. Alfalfa mosaic virus consists of 4 distinct particles. Most plant viruses have enough nucleic acid to code for only a very small number of proteins. Tobacco mosaic virus contains only 6,400 nucleotides, its one and only coat. Protein contains 158 amino acids, thus, a minimum of 158.3 is equal to 474 nucleotides are needed just for that. 3 other genes exist in this virus, 2 large ones code for replicase enzymes, and a smaller one codes for a protein thought to mediate spread of the virus from cell to cell. Most other plant viruses are similarly small. Virus metabolism. Viruses must always invade a living cell in order to reproduce, and all known types of organisms are attacked by viruses: plants, animals, fungi, protozoans, algae, and prokaryotes. Viruses that attack bacteria are called bacteriophages or phages, but these are viruses just like the others. Viruses that attack bacteria and animals are usually extremely specific. A particular type of virus can attack only a certain species of host. The same is true of many plant viruses, but others are able to attack many related plant species. Fungal and algal viruses are less well known. Figure 15.25. This diagram illustrates the method involved in using recombinant DNA techniques to genetically alter a plant. The method is explained in the text. Several components of a tissue culture experiment: the original plant, callus culture on agar in foreground, liquid cultures in flasks. The red liquid cultures have been experimentally induced to form pigments. The flask tipped on its side shows that these cells can grow so extensively that they consume all the liquid medium and form a solid mass of cells. Tissue culture experiments can also produce embryos. If they have been genetically engineered, they grow into altered plants. To invade a plant, viruses rely on damage to living cells, such as through the action of aphids, chewing insects, or open wounds left by pruning or breaking. If an insect has chewed or sucked on an infected plant, the virus particles adhere to its mouth parts and feet. If these penetrate a host cell, some virus particles are transferred, allowing a virus particle to enter the protoplasm directly, completely bypassing the cell wall. During experimental studies of virus activity, healthy plants are infected by first abrading the epidermis with sandpaper to create fine breaks in the cells and then rubbing into the wound a solution of virus particles extracted from an infected plant. Alternatively, freshly crushed, infected leaves are rubbed into the abrasion. After the virus is inside a suitable host cell, protein molecules of the coat fall away from each other, releasing virus nucleic acid. If the virus is a DNA virus, its liberated DNA is recognized by the plant's own RNA polymerases, and transcription of viral genes into mRNA begins. These mRNAs are translated by the plant's ribosomes, and within minutes of infection, new viral protein appears in the cell. Transcription and translation of some viruses are rather slow, and the virus does little harm. In other types, viral mRNAs dominate the ribosomes, and the plant's protein synthesizing apparatus is completely taken over. At the same time that RNA polymerases are transcribing viral DNA, DNA-replicating enzymes bind to and copy the DNA, making new viral double helixes. As the number of viral DNA molecules increases, the rate of viral transcription and translation also increases. Finally, all plant cell metabolism is redirected to viral metabolism. Retroviruses, which have RNA but no DNA, may act as an mRNA and be picked up by a plant ribosome and translated. Usually one of the first proteins to result is reverse transcriptase, which binds to the RNA and synthesizes a complementary molecule of DNA. Another enzyme then synthesizes the other half of the DNA duplex, and the rest of the steps are the same as for a DNA virus. Some retroviruses bring molecules of reverse transcriptase with them as part of their coat, therefore, reverse transcription is the first step. As virus components become more abundant, viral nucleic acid moves into surrounding cells by means of plasmodesmata, and perhaps by passing through the wall as well. Viral nucleic acid that enters phloem spreads rapidly. The only tissues that are somewhat safe are root and shoot apical meristems, which do not contain phloem. Even in a heavily infected plant, the shoot apex itself may remain virus-free. The rate at which viruses take over host cell metabolism is variable. In many plants, it is slow and viruses never become abundant. Such plants show few disease symptoms, and the presence of virus is detected only by electron microscopy. This type of virus is common in many crop plants. If the virus multiplies quickly and dominates the cell, symptoms such as chlorosis, necrosis, and leaf curling may appear. Such viruses are virulent. Figure 15.26. Turnip mosaic virus particles are long, narrow filaments. In this preparation, viruses have been isolated from plant cells, washed, and treated with heavy metals to make them visible in an electron microscope. Figure 15:27. Most viruses, but especially plant viruses, are extremely simple. Tobacco mosaic virus contains only one type of protein, which binds to the DNA, forming the long rod. In this form, the DNA is well protected. In the virus shown here, the protein does not bind so closely to the nucleic acid but rather forms a coat, often called a capsid, around the nucleic acid. Some viruses, once freed of their coat protein, insert viral DNA into the host DNA. Little is known about this process, but apparently, viral DNA lies next to host DNA, and then breaks are made in both molecules. The ends join, and the cuts are healed by DNA ligase. Such viral DNA usually lies dormant, perhaps simply because so little of the eukaryotic DNA is expressed in any particular cell. However, if this insertion happens in young plant tissues, viral DNA is replicated along with plant DNA during the S phase of every cell cycle, and both daughter cells receive infected DNA during mitosis. Such viruses are termed temperate and may produce no symptoms whatsoever. They may remain hidden forever and die when the plant dies. In some, however, they become active and reproduce just like virulent viruses. The factors that induce conversion from temperate to virulent are not known. Perhaps the viral DNA inserted itself downstream from the promoter region for a gene that is now being activated as part of the cell's differentiation. After the promoter region allows RNA polymerase to bind and transcribe, the viral DNA is transcribed as well. This hypothesis has not yet been proven, and other factors may be important. Figure 15:28. These cells of tobacco leaf are infected with tobacco mosaic virus. Although the cytoplasm is almost filled with virus particles, the surrounding cells appear healthy, and the vacuoles are virus-free. Formation of new virus particles. At some point, viral components assemble into new particles. For many viruses, such as tobacco mosaic virus, this is a self-assembly process. Viral coat protein has a tertiary structure that causes it to bind to viral DNA. This binding then permits it to attract and adhere to more viral protein, and a new protein DNA viral particle is quickly assembled. The protein monomers by themselves do not adhere to each other. Viral DNA must be present. No other components are necessary, and even in a test tube, the two self-assemble into infectious virus particles. In many types of animal viruses and bacteriophages, one of the last proteins made is an enzyme that destroys the host cell, causing it to burst and release virus particles into the environment. This does not happen with plant viruses. Perhaps the cell wall is too massive to be digested. Instead, virus particles remain in the cell until it is broken open by insects or larger animals. If an infected leaf is never eaten but instead is abscised in autumn, virus particles are released as the leaf decomposes. These too can be spread by animals. Origin of viruses. Many, if not most, viruses are actually portions of genes of the host species or a species closely related to the host. As living organisms are damaged or die in decay, their nuclei break down along with the rest of the cell material. It is possible that occasionally a fragment of a chromosome codes for proteins that can self-assemble into a crude coat and have some infectious potential. Such a fragment has the possibility of acting as the forerunner of a virus. The fragment does not have to be as efficient as a full-fledged virus because it can evolve just like any other genetic system. With a long span of time, natural selection favors mutations in this DNA fragment that improve its survival rate. Figure 15:29. Squash mosaic virus affects not only squash, but related plants as well, such as watermelon. Symptoms occur in both leaves and fruits. An alternative hypothesis postulates that viruses are highly evolved, extremely efficient parasites. Whenever an organism can obtain required compounds from its environment, it is selectively advantageous not to waste energy synthesizing them, but even the most reduced, simplified parasites are vastly more complex than viruses. Could viruses be reduced parasitic bacteria instead? This seems doubtful for most viruses because the metabolism of plant and animal viruses should then resemble bacterial metabolism, but instead, it matches plant or animal metabolism. Plant diseases caused by viruses. Plants suffer from at least 1,000 different virus-caused diseases. Because symptoms are similar to those of mineral deficiency or other environmentally caused problems, detection can be difficult. Few effective treatments exist for plants infected with viruses. We do not try to cure entire crops of viral disease, rather, we try to maintain healthy, uninfected breeding stock for virus-free seeds or cuttings. Heat treatment inactivates some viruses. Entire plants can be kept in hot growth chambers or greenhouses for several weeks to several months, after which they may be free of virus. Even these techniques are ineffective in most plants and against most virus diseases. The best policy is to use virus-free plants and protect them from infection. Virus-free plants are obtained by shoot meristem propagation or sometimes by normal sexual production of seeds, if stamens and carpels can grow and set seed more rapidly than virus particles can infect them. At the next level. 1. Proofreading and error correction during DNA replication. As enzymes copy DNA, they occasionally make mistakes by putting the wrong base in or by skipping several bases and not replicating them. Every error is a mutation. Many methods of deleting and correcting errors have evolved. Because plants grow by localized growth in shoot and root apical meristems, it is crucial for meristematic cells to detect and correct as many mutations as possible. As you investigate this, ask yourself what the consequence would be if an organism were to evolve such that it had perfect DNA replication, that either no errors were ever made, or if all errors could be detected and corrected. How would that affect the organism's further evolution? 2. Chromosome structure. In an introductory course such as this, we explain things at a basic level. A chromosome is presented as a single DNA duplex with a centromere and 2 telomeres. But as you might have guessed, chromosomes are much more complex. Only very small regions of the DNA consist of genes. Other parts are control elements where regulators, activators, or inhibitors attach. There are regions that allow DNA to become attached to chromosome structural proteins and to the skeletal proteins of the nuclear envelope, and so on. All these aspects of DNA affect the expression of genes, and all are important in plant development. 3. DNA methylation and DNA imprinting. For most traits, it does not matter which allele of a gene we inherit from our mother and which from our father. The same is true for plants. But for a small number of traits, it matters very much which allele comes from which parent. As it turns out, in both plants and animals, eggs often modify some of the genes by adding methyl groups to some of the bases, whereas the sperm cells do not. Once combined into a zygote, the mother's genes have more influence than do the father's. The consequences and benefits of this are not yet completely understood, and now we have just discovered that in humans, fathers also mark some of their genes. It is possible that DNA imprinting will be one of the most important topics in developmental biology in the next several years. 4. Plant cloning through tissue culture and embryogenesis. It is now extremely easy to modify DNA by deleting certain elements or adding new parts. In many cases, the difficulty is then inserting the new DNA into a plant cell and forcing that cell to grow into a new plant. Often, it will grow only as an irregular lump of callus in tissue culture. A great deal of research has been aimed at developing tissue culture techniques that could easily and reliably cause altered cells to grow into plants. That goal has not been achieved, but every year some progress is made. If we could trick a modified cell into acting like a zygote, the process might be universally effective. Summary. 1. All information required to specify protein primary structure, the sequence of amino acids, is stored as the sequence of deoxyribonucleotides in DNA. 2. Cell differentiation is based largely on differential activation of genes and control of the processing of heterogeneous nuclear RNA into messenger RNA. 3. The exact details of the mechanism by which a plant hormone induces differential activation of either nuclear or organellar genes are not known. Binding of a hormone to its receptor results in the formation of transcription factors that bind to DNA promoter regions. 4. The genetic code consists of triplets of nucleotides, each triplet coding for only one amino acid, or for stop or start. The code is degenerate, each amino acid being coded by several codons. 5. Genes consist of a promoter region that contains enhancer elements and a structural region that usually contains both exons and introns. 6. In transcription, RNA polymerase attaches to the promoter region, moves to a start site, and then polymerizes RNA, being guided by base pairing in a short region of single-stranded DNA. Both introns and exons are transcribed. 7. Heterogeneous nuclear RNA is processed to mRNA and then transported to the cytoplasm, where it binds to ribosomes. Each ribosome has a large and a small subunit, 4 molecules of rRNA, and approximately 80 proteins. 8. Amino acids are carried to ribosomes as part of an activated tRNA, each of which has an anticodon complementary to the codon for the amino acid it carries. All tRNAs have similar structures. 9. Restriction endonucleases cut DNA at specific sequences. The resulting pieces can be melted to the single-stranded state. Single-stranded nucleic acids from different sources can be mixed and allowed to hybridize, either as a measure of their relatedness or as part of the construction of a new molecule of DNA. 10. Specific sequences of DNA can be synthesized artificially by incorporating one copy into a vector and then inserting the vector into a bacterium. As the bacterium reproduces, the sequence of DNA is reproduced as well. 11. Most viruses are short pieces of DNA or RNA that contain a few genes closely related to normal host genes. Most plant viruses have RNA, not DNA, and a code of just one type of protein. 12. Viruses infect plants through wounds and then divert the plant's nucleic acid and protein synthesizing metabolism to the synthesis of more virus molecules, which then self-assemble into complete virus particles. Important terms. Anticodon bacteriophages, chromatin codons. Complementary DNA differential activation of genes, DNA cloning, DNA denaturation, DNA hybridization, DNA ligase, DNA microarray. Eukaryotic initiation factors, exon. Expression profiling, gene introns. Messenger RNA palindromes. Polymerase chain reaction promoter region recombinant DNA techniques, restriction endonucleases, restriction map, retroviruses, reverse transcriptase, ribosomal RNA, ribosomes, start codon, stop codon. Structural region transcription. Transfer RNA. Yeast artificial chromosome. Review questions. 1. Plants are composed of numerous types of cells that are all unique because they have distinct metabolisms. What are these metabolisms based on? 2. The information needed to construct each type of protein is stored in. 3. Because an organism grows by duplication division, all its cells have genes. 4. What is meant by the differential activation of genes? Explain how this affects the synthesis of cutin and P protein. 5. Cutin, lignin, and chlorophyll are not proteins. How is it possible for genes to control the synthesis of these polymers? 6. A gene is made up of. 7. For each of these symbols, write out the full name of the base and of the nucleoside. Indicate which are components of DNA and which are components of RNA, A, TB. A, C, G, D, C, E, U. 8. The text describes 3 important ways that cells protect their genes. List these methods. 9. DNA does not participate directly in the synthesis of proteins. What molecule transfers the information in DNA to the site of protein synthesis? 10. Histones are proteins in the nucleus, and DNA wraps around them, forming a spherical structure. What is the name of the structure? 11. What is the name of the enzyme that digests DNA? 12. What is a codon? How many exist? Why is the genetic code described as being degenerate? 13. Examine Table 15. 2. Which amino acids are coded by the following codons? A, UUB, UCUC, UAUD, UGCE, CUUF, CJG, CGG. H, AAI, AGG, GAGK, GGUL, GGCM, GGN, GGG. 14. What is unusual about the answers for questions 13K, 13L, 13M, and 13N? Because the multiple codons exist for most amino acids, the genetic code is said to be 15. Examine Table 15. 2. What do the following codons specify? A, UAB, UGC, UAG. What happens when a ribosome comes to one of these codons? 16. Examine Table 15. 2. The codon August codes for the amino acid methionine and also for start. What does start mean? 17. What are the two portions of a gene? Which portion actually codes for the amino acid sequence? 18. Describe the promoter and structural region of a nuclear gene. What is a Tata box? What are exons and introns? 19. The structural portion of genes contains two distinct types of regions, exons and introns, which consists of codons that are eventually translated into the amino acid sequence of a protein, and which consists of codons that are not expressed. 20. After RNA polymerase binds to DNA, it begins making mRNA. What is the name of this process? 21. What is the name of the cytoplasmic particles that translate mRNA into proteins? What type of RNA brings amino acids to this particle? This RNA has a region called an anticodon. What does the anticodon do? 22. One of the codons in mRNA that specifies the amino acid phenylalanine is UUC. What is the anticodon on the tRNA that carries phenylalanine? 23. What is a frameshift error? Does the binding of methionine tRNA to the small subunit help reduce frameshift errors? If so, how? 24. If a molecule of mRNA had the following sequence, what would be the sequence of the gene that coded the mRNA? Which amino acids would be incorporated into the protein? Gene, 3'-5. mRNA, 5' UCGACAUUGUCCCGGCCUC3 protein. 25. Describe the steps of polypeptide elongation by ribosomes. 26. When a stop codon is pulled into the A channel of a ribosome, what happens? Is there any tRNA with an anticodon that is complementary to the stop codon? 27. There are three main classes of RNA involved in protein synthesis: RNA, RNA, and RNA. 28. Describe DNA melting and DNA reannealing. 29. What is a restriction endonuclease? 30. What are palindromes, and how are they related to restriction endonucleases? Why are they useful for inserting one piece of DNA into another? 31. DNA fragments can be identified. For example, if plastid DNA is treated with PST-I restriction endonuclease, it cuts the DNA into about 10 to 20 fragments. If these are separated on an electrophoresis gel and then stained, this is A of the plastid DNA. 32. Imagine that plastid DNA of species A is treated with a restriction endonuclease in order to make a restriction map. Now imagine that species B is not very closely related to species A, but species C is. If plastid DNA of species B and species C are treated with the same restriction endonuclease that was used for species A, will the restriction map of species B or species C be more similar to that of species A? 33. If mRNA is extracted from a plant and mixed with reverse transcriptase, what is the name of the DNA that is produced? If it was synthesized with radioactive nucleotides, what can be done with it? 34. Briefly describe how a piece of DNA can be cloned using bacteria. Why would it be important to use the same type of restriction endonuclease in the harvest phase as in the preparation phase? After the DNA had been cloned, how would you go about sequencing it? 35. Briefly describe how a piece of DNA can be cloned using PCR. 36. In the genetic engineering of plants, after a gene and promoter have been prepared, they must be inserted into the plant with an insertion vector. A very important vector comes from Agrobacterium tumefaciens. What is the vector's name? After the vector and the gene plus promoter have been added to many plants, the plants are treated with a herbicide. Why? Which plants will survive? 37. Corn has been genetically engineered to produce the anti-insect poison of a bacterium Bacillus thuringiensis. What is one benefit of this? What are some of the risks? 38. Viruses are extremely small particles that usually contain only and. 39. The diversity of nucleic acids in plant viruses is great. Which class of viruses contains single-stranded RNA? These viruses are the source of which important enzyme? 40. There are 3 hypotheses about the origin of viruses. Briefly describe each hypothesis. 41. About how many plant diseases are caused by viruses. Design credits: Hummingbird, Tango 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated Z-Art. Part 3. Genetics and Evolution. Chapter 16 Genetics. Chapter 17 Population Genetics and Evolution. Chapter 18 Classification and Systematics. Chapter 19 Algae and the Origin of Eukaryotic Cells. Chapter 20 Non-vascular plants, mosses, liverworts, and hornworts. Chapter 21 Vascular plants without seeds. Chapter 22 Seed plants I, Gymnosperms. Chapter 23 Seed plants 2, Angiosperms. Chapter 24 Ethnobotany. Part opener image. Legumes are rich in protein and have been an essential part of the diet of human beings for thousands of years. During that time, people noticed differences in the seeds they harvested. Some tasted better, some had attractive colors, some seemed to satisfy hunger longer. In addition, they searched for plants that seemed similar to the legumes they knew, hoping to find new types of beans, peas, and lentils. These early people cultivated the seeds they liked the best and discarded others. Gradually, a wide variety of legumes entered our diet. Selection like this, performed by people, is artificial selection, whereas selection that occurs in nature is natural selection. Both result in evolutionary change. The focus of this part is the mechanism by which DNA and its information are passed from parent to progeny, how that DNA changes over time, resulting in new types of organisms, and how the environment and natural selection have interacted to produce hundreds of thousands of species of prokaryotes, algae, fungi, animals, and plants. DNA serves as an archive of information that, interacting with environmental information and resources, produces an organism's body, its structure, metabolism, and biology. DNA also carries the information needed to make more DNA so that during reproduction, either of the cell or of the entire organism, each progeny receives copies of the DNA and the information it contains. Errors occur occasionally, and molecules of DNA are not copied perfectly. Each version may code for slightly different enzymes or in some other way produce an organism slightly different from parents or siblings. If the world were absolutely uniform, we might expect that whenever a mutation resulted in a new type of individual, either the new type or the original would be favored by natural selection until it was the only type left. A more advantageous metabolism or structure would be more advantageous everywhere. However, our world is complex, containing aquatic and terrestrial environments, areas with mild temperatures and others with temperature extremes, and a diversity of rainfall, soil types, pathogens, pollinators, and so on. Consequently, alternative biologies may each be selectively more advantageous in different conditions. The diversity and richness of the environment result in diversity and richness of organisms. Chapter 16. Genetics. Chapter opener image. As this bee gathers nectar and pollen from these flowers of Nolina perryi, it is setting up thousands of experiments in genetics. It carries pollen grains from some plants and deposits them on the carpels of other plants. Because of the crossing over that occurs in meiosis, none of the sperm cells is identical to any other in the alleles they carry, and each egg also differs from all others. Therefore, each seed that results from this bee's activity will differ genetically from all other seeds, and some will be more adapted to this environment than others. Chances are good that those that are more adapted will grow and reproduce better than those that are poorly adapted. Outline. Concepts. Replication of DNA. Mutations. Causes of mutations. Effects of mutations. Somatic mutations. DNA repair processes. Monohybrid crosses. Monohybrid crosses with incomplete dominance. Crossing heterozygotes with themselves. Monohybrid crosses with complete dominance. Test crosses. Multiple alleles. Dihybrid crosses. Genes on separate chromosomes, independent assortment. Crossing over. Genes on the same chromosome, linkage. Multiple genes for one character. Other aspects of inheritance. Maternal inheritance. Lethal alleles. Multiple sets of chromosomes and gene families. Box 16 to 1 Botany and Beyond, Botanical Philosophy and Popular Culture. Box 16 to 2 Botany and Beyond, Whose genes do you have? Box 16 to 3 Alternatives, Genetics of haploid plants. Learning objectives. After reading this chapter, students will be able to explain the advantage of sexual reproduction over asexual reproduction. Discuss DNA replication. State the importance of transposable elements in DNA mutations. Recall why DNA repair mechanisms are necessary. Describe the process of making and analyzing a monohybrid cross with selfing. Explain the purpose of a Punnett square. Discuss how a test cross can reveal a plant's genotype. Describe a complication of performing a dihybrid cross. Compare independent assortment to linked genes. Give an example of epistasis. Compare biparental inheritance to maternal inheritance. Discuss triploid and tetraploid cells. Did you know? Plants inherit one set of chromosomes through the sperm cell in pollen and one set through the egg cell in the ovule. Plants are promiscuous, each plant will have hundreds or thousands of flowers, and it is possible that pollinators will bring pollen from so many other plants that each seed has a different father. All the seeds produced by a single plant have the same mother, the plant itself. Domesticated crop plants have been interbred with each other for thousands of years, but most still have wild relatives that can interbreed with them. Concepts. Genetics is the science of inheritance. The chemical basis of genetic inheritance is the gene, the sequence of DNA nucleotides that guides the construction of RNA and proteins and also controls construction of more copies of the genes themselves. If all plants of a species had exactly identical nucleotide sequences in their DNA, then all those plants would be identical physically, but virtually all genes occur in multiple forms known as alleles, the alleles of a particular gene differing from each other in their sequence of nucleotides. Alleles arise by mutation. If the nucleotide sequence is changed in any way, the new sequence is a new allele. Mutations can occur in any gene in any individual, and thus, gradually a population of separate plants comes to have a variety of alleles. The types of alleles that a single individual has are called its genotype, and the expression of those alleles in the individual's size, shape, or metabolism is its phenotype. As a result of mutations, the population of individuals has varying genotypes and phenotypes. They are not identical, as is apparent from considering humans. An important concept of inheritance is the selective advantage of reproduction, which may be either sexual or asexual. In asexual reproduction, each offspring is identical to its parent and siblings, having exactly the same DNA and thus the same alleles. Although this might seem like the safest, most efficient mechanism for producing large numbers of offspring to carry the parent's genes into future generations, the progeny are never more fit than the parent, and no progeny is adapted to any environment other than the parent's environment. During a drought, plants with poor capacity to withstand water stress are outcompeted by plants whose alleles confer superior water stress resistance. During an infestation of pathogenic fungi, individuals without alleles that confer immunity are destroyed. There may be no individual resistant to both stresses. Figure 16, one flower color in 4 o'clocks is controlled in part by a gene that has 2 alleles. The DNA sequence of one results in a protein whose primary and tertiary structures cause it to synthesize red pigment. The DNA sequence of the other allele codes for a protein whose tertiary structure is misformed, it has no enzymatic activity, and no pigment is produced. Therefore, flowers are white. If only a small amount of pigment is produced, the flower is pink. Flower color is the plant's phenotype, the type of alleles present is its genotype. Figure 16, 2. A population probably contains a diversity of genotypes and phenotypes. Without sexual reproduction, selectively advantageous alleles of one individual cannot be combined with those of another, and multiple stresses might kill all individuals. With sexual reproduction, traits of one individual are combined with those of another, often producing individuals more fit than either parent. Sexual reproduction is a mechanism by which an organism combines its alleles with those of other, possibly better adapted individuals, thereby increasing the probability that copies of its own alleles survive. If a plant is susceptible to drought, sexual reproduction is advantageous because some of its sex cells may fuse with those of a drought-resistant plant. If so, the new zygote should grow into an individual resistant to water stress, thus adding some protection to the alleles derived from the susceptible parent. Although the original plant may die during a drought, copies of its alleles survive in its progeny, protected by alleles from the resistant parent. The second parent should benefit as well, as it is almost certainly not superior in all attributes. The drought-sensitive parent may carry alleles for resistance to fungal attack, which, during times of fungal attack, protect the alleles of the drought-resistant parent in the offspring, making the progeny of this sexual reproduction resistant to both fungi and water stress. Sexual reproduction involves a large degree of chance and risk. Fitness and survival capacity are not governed by just 1 or 2 genes, but by almost all genes, including those responsible for the proper construction of membranes, the functioning of organelles, the production and transport of hormones, and so on. One cannot say that any aspect is trivial and does not matter, therefore, the presence of 1 or 2 particular alleles in a sex cell is not the key feature, but rather the combination of all the alleles. Because of synapsis and crossing over in meiosis, each plant produces thousands of types of sex cells, each with a unique genotype, which then fuse with the sex cells of many other individuals. For example, when plants bloom, they produce thousands or millions of pollen grains that are carried away and fertilize ovules on many other plants. Simultaneously, its own ovules are receiving pollen from numerous plants. As a result, thousands of fertilizations may occur that involve this one plant's alleles. Many may produce poor combinations of alleles that have little survival value, but at least a small percentage should have the best attributes of both parents and should produce healthy, genetically sound plants with a high capacity to survive all stresses. It is not necessary or even advantageous for all potential progeny to survive, only the fittest. The plants and animals around us today are the successful survivors of evolutionary experimentation. Figure 16, 3. Being diploid like most plants, this lupin has one set of chromosomes from its paternal parent and one from its maternal parent, but the meiosis that precedes formation of each pollen grain and each ovule ensures that none of its many gametes is the same as any other. Insects will carry pollen to this plant from many other lupins, some of which may be better adapted than this plant is. Replication of DNA. Before a cell undergoes nuclear division, either mitosis or meiosis, DNA is replicated during S phase of the cell cycle. Replication doubles the amount of DNA, and each gene exists in at least 2 copies, one on each of the 2 chromatids, one of which goes to each daughter nucleus during anaphase. As DNA replication begins, chromatin first becomes less compact, opening sufficiently to allow entry of the necessary replicating factors. DNA does not release from histones, instead, nucleosome structure remains intact. Next, one strand of the DNA double helix is cut, and the 2 strands separate from each other in a short region, forming a small bubble called a replicon. With the double helix open, free nucleotides diffuse to regions of single-stranded DNA and pair with its bases along both strands. These are ribonucleotides, not deoxyribonucleotides, and they are polymerized into short pieces of primer RNA approximately 10 nucleotides long. The primer RNAs then act as substrates for the DNA synthesizing enzyme, DNA polymerase. It now enters and adds deoxyribonucleotides onto the end of the primer RNA using the open DNA as a guide. This method of replication, in which each strand of DNA acts as the template for making the complementary strand, is semi-conservative replication because each resulting double helix contains one new molecule and has conserved one old one. DNA polymerase adds deoxyribonucleotides only to the 3' end of the growing nucleic acid, and thus, one strand of the open DNA is copied in one direction and the other strand in the opposite direction. As DNA continues to unwind and open in both directions, new pieces of primer RNA form and new pieces of DNA grow from them toward the existing fragments. The original primer RNAs are depolymerized, and new Okazaki fragments are joined to the first ones. On the other end of each strand, replication is continuous, one long chain forming from the original primer RNA. Each chromosome contains only a single DNA double helix, and in each chromosome, hundreds or thousands of sites occur where replication is initiated. As DNA uncoils and then separates, it has a forked appearance. This region is a replication fork. As DNA continues to uncoil and open, replication forks at each end of each replicon travel along the DNA. Finally, each replication fork runs into one of the adjacent replicons, then all new pieces of DNA are ligated into two new, complete molecules. The large number of initiation sites allows numerous DNA polymerases to work simultaneously. Each is able to incorporate approximately 1,000 nucleotides every second into growing DNA fragments, and replicons extend at the rate of about 0.5 mm min. Each replicon is about 45,000 to 180,000 base pairs long and can be replicated in about 1 to 3 hours. Because S phase of the cell cycle is 3 to 10 hours long, only about 1/3 to 1/10 of the replicons are active at any particular moment. If only one DNA polymerase could act on each chromosome, S phase would require 40 to 60 days. As replication forks advance, DNA partially dissociates from the histone octamers, but as all enzymes migrate forward and the two new DNA double helixes are complete, they immediately reassociate with histones into complete nucleosomes. Some of the old histone octamers go to one double helix and some to the other, and new histone octamers are added to both. Any particular segment of DNA remains unpackaged for only a few moments. Figure 16, in order for DNA to unwind, one strand must be cut, otherwise, the entire unreplicated part would have to spin rapidly, which is impossible. With a single cut, only the region between the replicating enzymes and the cut must rotate. Figure 16, after a replicon opens, two pieces of primer RNA are formed, then DNA polymerase adds new DNA to the 3' end of the primers, and both strands elongate as the replicon unwinds at both ends. As the replicon continues to open, Okazaki fragments are initiated periodically, they grow to the downstream fragments, and the enzyme DNA ligase joins them into single molecules. Table 16, 1 amount of DNA per haploid set of chromosomes. The values are known for only a few species at present, but more are being analyzed. Diploid nuclei have twice as much as shown in this table. Mutations. A mutation is any change, however large or small, in DNA. The smallest mutation, affecting the least amount of DNA, is a point mutation in which a single base is converted to another base by any of various methods. If a piece of DNA is lost, the mutation is a deletion, the addition of extra DNA is an insertion. Under some conditions, a piece of DNA becomes tangled and breaks, and during repair, it is put in backward as an inversion. Figure 16, consider this initial sequence of DNA, the box is not important. A point mutation consists of the change in just a single base pair, here GC is converted to AT. If this sequence is part of a gene rather than just spacer DNA, this change in sequence has resulted in a new form of the gene, a new allele. The 6 base pairs in the yellow box of the initial sequence have been deleted. Deletions often remove hundreds of base pairs. On the upper strand, the right boundary of the deletion has removed a C and an A and left a T, changing cat to AT. The August mRNA start codon is coded by the DNA triplet cat. Therefore, this may be an important mutation, destroying a start codon, but this may not be a triplet. The real triplets may have been GCATA or TGCATA. The diagram provides too little information to predict the significance of the mutation. An insertion is the addition of one or more base pairs. In this inversion, the DNA has broken at 2 points and then flipped over and been reinserted. The original top strand of the insert is now the bottom strand. Causes of mutations. A mutagen is something that causes mutations. Several that are important are certain chemicals, ultraviolet light, X-rays, and radiation from radioactive substances. Many mutagenic chemicals are man-made and are increasing in our environment. One mutagen, nitrous acid, reacts with cytosine and converts it to uracil, giving the DNA a GU base pair in place of a GC base pair. Numerous classes of DNA repair enzymes exist, one of which recognizes this GU pair as abnormal and changes it back to GC, but in meristematic cells that are replicating their DNA, DNA polymerase may arrive before the repair enzyme. If so, one new strand is formed with a complementing the U. In later replication, the A guides the incorporation of thymine, and the original GC base pair becomes AT. Deletions can be caused in several ways. One method is due to short regions of a self-complementary sequence. As DNA unwinds ahead of the DNA polymerase, one strand with a self-complementary sequence may form a small loop. DNA polymerase may pass by this loop without reading any of the bases in it. The bases complementary to those in the loop are left out of the new DNA molecule, so it is shorter than it should be. Figure 16, 7-nitrous acid converts cytosine to uracil. This strand is mutated, and all strands derived from it will be mutated, having AT base pairs. The guanine is not affected and produces the original allele. One new allele has come into existence. Insertion mutations can be caused by many methods because a variety of enzymes cut and rejoin DNA as part of repair processes. If a small piece of foreign DNA is present after cutting, it may accidentally be incorporated into the chromosomal DNA by DNA ligase. Transposable elements are one of the most interesting and useful causes of insertion and deletion mutations. These are pieces of DNA that readily change their positions from one chromosome to another. Transposable elements have two basic forms, insertion sequences and transposons. Insertion sequences are only a few thousand base pairs long and contain the genes that code for the enzymes actually involved in cutting the insertion sequence out and splicing it into DNA somewhere else. A transposon is like an insertion sequence except that it may be much longer and carries genes that code for proteins not associated with transposition. The deletion and insertion mutations caused by transposable elements can vary in severity. If the element inserts into spacer DNA, the effect is not very important, but if it inserts into a gene, it totally disrupts either the promoter or the structural region. We suspect that transposable elements may actually be viruses, that they are a disease. Plants have mechanisms that fight them, but most plant nuclei contain transposable elements, sometimes in large amounts. More than half the DNA in every nucleus of Norway spruce is transposon DNA, less than half of each nucleus is the plant's own DNA. Mutations caused by transposable elements may be one of our most powerful tools for genetic analysis and engineering. Often, a metabolic pathway is studied by exposing thousands of plants to mutagens such as X-rays or nitrous acid, then examining the plants to find any that have mutations disrupting the metabolic pathway of interest. These are then studied further to determine how the pathway was affected, but we never actually know which gene was disrupted. The gene is given a name, but we do not know its DNA sequence, its location on a chromosome, or the nature of its promoter or structural regions. However, transposons are now being sequenced and engineered to contain markers such that the transposons can be applied to plants or cultured cells, where they cause insertion mutations. The mutants of interest are located, and DNA from the mutants is cut into pieces by restriction endonucleases, denatured, and combined with radioactive DNA complementary to the transposon. After the piece is found, it can be sequenced. Because we know the transposon sequence already, the DNA on either side of the transposon must be the gene of interest. Transposon mutagenesis offers the possibility of quickly correlating metabolic pathways, proteins, genes, and even promoters for virtually any aspect of an organism. Table 16, 2 names of genes in pea plants. Although there are thousands of alleles in plants, only a few ever become obvious, and only a small number of those are ever studied and given names. These are just a few of the named alleles in pea. Figure 16, 8 transposon mutagenesis. The method is described in the text. In this case, flower pigment synthesis is of interest. Plant 1 is a normal plant. Plant 2 has a mutation for leaf shape, which is not our objective, and plant 3 has white flowers. Thus, we have induced a mutation in some gene that is necessary for pigment synthesis. Effects of mutations. The effect and significance of a mutation depend on its nature, its position, and its extent. If it occurs in spacer DNA between 2 genes, it may have no effect whatsoever. Also, point mutations and small insertions and deletions in introns appear to be unimportant generally. They change a portion of hnRNA that will not be incorporated into mRNA. Within exons, a small mutation may not be important if it only changes a codon into another codon that specifies the same or a similar amino acid. Changing codons to ones that specify very different amino acids may not matter if they are located in a part that is not critical to the protein's functioning. On the other hand, a mutation in an exon may cause the gene to code for a protein whose active site is disrupted and the protein cannot function. An insertion mutation may cause the gene to code for a protein so long that it cannot fold properly. Also, mutations in promoter regions can completely inactivate a gene or cause it to be active at the wrong time or place. Even point mutations can have profound effects. For example, nitrous acid conversion of a GC base pair to AT might cause the formation of a new start codon. Other simple changes can eliminate start codons, the recognition site for distinguishing the boundaries of exons and introns, tauta boxes, and so on. The larger the mutation is, the greater the probability that a critical part of the DNA is affected. Statistically, mutations are almost always harmful. Enzymes tend to be approximately 300 to 400 amino acids long, and hundreds of trillions of proteins could possibly exist. Yet only a small fraction would be useful in living organisms. Any mutation that changes the structure of proteins, rRNA, or tRNA is more likely to produce a less useful than a more useful form. The majority are deleterious, the minority beneficial. Natural selection eliminates the deleterious mutations and preserves the beneficial ones. Somatic mutations. Mutations can occur at any time in any cell, but if they happen in cells that never lead to sex cells, they are called somatic mutations. For example, a gene in a leaf primordium cell may undergo a mutation, but because leaves are not involved in sexual reproduction, the mutation is somatic and is not passed on to the plant's offspring, regardless of whether the mutation is advantageous or disadvantageous. When the leaf falls off in autumn, the mutated gene decomposes along with the rest of the leaf. The same is true of any mutation in roots, wood, or bark. In general, somatic mutations are not very important for most plants because they affect such a small portion of the plant and are not passed on to the offspring. A somatic mutation might not ever result in an altered phenotype. The mutation may occur in a leaf cell nucleus, but in a gene that is inactive in leaves, such as a gene that affects root hair growth or bark formation. In species that undergo extensive vegetative reproduction, such as blackberries, alder, and prickly pear cactus, somatic mutations can be important if they affect a part of a plant that gives rise to a vegetative offshoot. As the offshoot grows and reproduces vegetatively, the patch of mutated cells may finally include an axillary bud and then affect a flower and its sex cells. The mutation then becomes a sexual one. DNA repair processes. Because most mutations are deleterious and occur frequently enough to be a significant problem, it is selectively advantageous for organisms to have DNA repair mechanisms that recognize and remove mutations. Certain mechanisms recognize base mismatches, loops, or other problems. Other enzymes minimize the number of errors that occur in the first place. The DNA repair rate must be neither too efficient nor too ineffective. In organisms that have very short genomes, such as prokaryotes and perhaps algae and protozoans, DNA polymerases can replicate a full genome without errors. Perfect replication does not occur every time, but non-mutated replication can sometimes occur. With the larger genomes of more derived animals and plants, one set of chromosomes can virtually never be replicated without mutations if no repair mechanisms are present. DNA polymerase may make an error only once in every 1 million nucleotides, but if the genome is several billion base pairs long, every replication produces numerous errors. Most angiosperms have about 10 to 100 billion base pairs per haploid set of chromosomes. We humans have only 3 billion base pairs. One of the smallest numbers known is an Arabidopsis, with about 120 million base pairs. Every round of replication before a mitotic or meiotic division results in about 120 errors. As a zygote grows into an adult, every cell cycle introduces new mutations, and in the adult, no two nuclei are exactly alike. After several cell cycles, DNA would be useless if there were no repair mechanisms. It would be impossible to produce sperms or eggs that were not extensively mutated, and under such circumstances, complicated organisms could not exist. It is estimated that a minimum of 15,000 genes are needed to code for all the information required for a flowering plant. This is about 20 million base pairs, so DNA polymerase is not accurate enough to provide error-free replication for even the simplest angiosperm, but DNA proofreading and repair systems bring the error rate down to an acceptable level. Actual rates as low as 1 mutation per 500,000 genes have been measured in corn. Mutations occur in the genes that code for repair enzymes, resulting in serious problems. In humans, the disease xeroderma pigmentosum is caused by an inability to repair mutations caused by ultraviolet light. People with this disease are sensitive to sunlight and develop skin cancers easily. Sunlight has the same effect on all of us, but most of us can repair the damage. Our bodies have mechanisms that repair other types of mutations as well, but our modern chemical society may be contaminating our environment with mutagens for which we have no repair mechanisms. DNA repair mechanisms must not be perfect or even extremely efficient. If every replication were perfect and if no mutations ever arose, all cells of an individual would have absolutely identical nuclei, as would all sperm and egg cells. Sexual reproduction would be useless because all eggs and sperms of a species would carry identical genes. Mutations must occur at a low enough rate that they do not endanger every individual, but rapidly enough that a species evolves as its environment changes. With no variation, there would be no differences for natural selection to act on. If all are identical, none has a selective advantage. Monohybrid crosses. Sexual reproduction between two individuals is called a cross. The meiotic divisions that precede a cross reduce the number of sets of chromosomes per cell from the diploid number to the haploid number. Consequently, each sex cell, that is, each sperm cell and egg cell, contains one complete set of genes. Furthermore, each sperm cell contains all the genes necessary to construct a new plant. The same is true of each egg cell. The zygote has two complete sets of genes. Within a population, mutations produce new alleles, and the genotypes of individuals within the population differ. Of the plants that grow in an area and that can interact sexually, many may have the same allele of a particular gene, but other individuals may have other alleles, other versions of the gene. Consequently, the alleles carried by a particular sperm cell may or may not be identical to the homologous alleles of the egg it fertilizes. Monohybrid crosses with incomplete dominance. In a monohybrid cross, only a single character is analyzed and studied. The inheritance of other traits is not considered. For instance, a plant with red flowers might be crossed with one that produces white flowers, and only the inheritance of that flower color trait is studied. Characters involving flower shape, leaf structure, and photosynthetic efficiency are also being inherited simultaneously, but in a monohybrid cross, only one is studied. This makes the analysis and understanding of the results much simpler. After basic principles of a particular trait are known, then its interaction with another factor or two other factors can be studied. Mendel gave his attention to monohybrid crosses, which are easy to understand. Before him, people tried to analyze many characteristics at a time and became hopelessly confused. Consider the cross just mentioned. A plant with red flowers is bred to one with white flowers. It does not matter which flower produces pollen and which produces ovules. After the cross is made, seeds and fruit develop, the seeds are planted, and when mature, the new plants are allowed to flower so that their flower color can be examined. The flowers of all plants in this new generation are pink, resembling each parent somewhat, but not exactly like either. The parents are called the parental generation, the offspring of their crossbreeding are the F or first filial generation, and if these interbreed, their offspring are the F generation. The molecular biology of this monohybrid, flower color cross is easy to understand. Each parent is diploid and thus has two copies of the gene involved. In the red-flowered parent, both alleles produce mRNA that is translated into functional enzymes involved in synthesis of red pigment. In a white-flowered parent, both alleles are defective. It may be that each produces an mRNA that when translated results in a protein unable to perform the necessary reaction, or the promoter region may be mutated and can no longer interact with a chemical messenger. Whatever the cause, there is no pigment, and the flower is white. Using genetic symbols, the red-flowered parent is RR and the white-flowered one is RR. Each parent is said to be homozygous because each has two identical alleles for this gene. The pink-flowered F has received an R allele from one parent and an R allele from the other, so its genotype is RR. It is heterozygous because it has two different alleles for this gene. We use these symbols even though we have neither isolated the gene nor analyzed its nucleotide sequence. R and R are simply labels. Most genes are known only by their phenotypes and the labels given to them by geneticists. With a genotype of RR, the plant produces mRNA, half of which carries the defect, thus, only half the normal amount of enzyme is produced. This results in less pigment being formed, only enough to make the flower look pink, not red. Neither parental trait dominates the other, so this pair of alleles shows incomplete dominance. The heterozygous phenotype differs from both homozygous phenotypes. When analyzing the possible outcomes of crosses and breeding, one must understand the types and quantities of gametes involved. All of the plants we are considering are diploid and form haploid spores by meiosis. The RR parent has chromosomes as shown in Figure 16, 9b, and regardless of how chromosomes separate during meiosis, all spores receive an R allele. In the RR parent, all spores receive an R allele. Spores are not sex cells, but as they develop into gametophytes, they divide by mitosis, duplication division, so all cells of the microgametophyte have the same allele as do all cells of the megagametophyte. Because the RR parent produces only R spores, it also produces only R sex cells, both sperms and eggs in typical bisexual flowers. Similarly, the RR parent produces, indirectly, only R gametes. When the two plants are interbred, an R gamete unites with an R gamete, establishing a heterozygous zygote that grows into a heterozygous adult by means of mitotic cell divisions. No other outcome is possible. It does not matter which gamete is which, an R sperm and an R egg result in the same type of zygote as an R sperm and an R egg. When the heterozygote matures and flowers, each spore mother cell produces two types of spores, not just one as is true of a homozygote. Because each cell has one R allele and one R allele, during the first meiotic division, one daughter cell receives R, and the other receives R. The second division of meiosis results in two R spores and two R spores. If this is occurring in the anther, half the pollen grains and hence half the sperms have R, and the other half have R. In the nucellus of many plants, only one megaspore survives. If this gene has no effect on spore metabolism, then half the time our cells survive and half the time our cells live. In heterozygote parents, the two types of sperms and eggs are produced in equal numbers, crossing heterozygotes with themselves. When a plant's own pollen is used to fertilize its own eggs, the cross is a selfing. A plant can also be selfed by being crossed with another plant with exactly the same genotype. Selfing heterozygotes has interesting, instructive consequences. 50% of all sperms and eggs contain the R allele, and 50% have R. Not all zygotes are identical genotypically, some are RR, having resulted from an R sperm and an R egg, others are RR, and some are RR. Selfing a heterozygote produces three types of FS, some of which resemble the parents and others the grandparents. Again, to analyze the results, we must wait for the zygote to develop into an embryo, then plant the seeds and wait until the new plants are old enough to flower. Because each genotype produces a distinct phenotype, the genotype of each plant is known simply by looking at the flowers. If a large number of heterozygotes are selfed and large numbers of F plants grown, approximately 1/4 are red, 1/2 pink, and 1/4 white. This is an important ratio, typically represented as 1:2:1, and should be memorized immediately. The reason for the proportion of these genotypes is explained in Figure 16:11. A Punnett square can be set up in which all types of one gamete, say, the egg, are arranged along the top of the square, and all types of the other gamete are arranged on the left side. The boxes are then filled in with the allele symbol above it and to the left. Because the gametes are produced in a ratio of 1R, listing them as in Figure 16:11 automatically represents their relative numbers in nature. Figure 16:9, a monohybrid cross analyzing the trait of flower color. Details are explained in the text. The phenotypes of parents, gametes, and progeny. Figure 16:10, production of haploid cells by meiosis in a heterozygote. Homologous chromosomes pair during metaphase I, then the paternal chromosome is separated from the maternal chromosome during anaphase I. One type of allele is separated from the other. As sister chromatids separate after metaphase II, each daughter nucleus receives identical alleles. The Punnett square does not represent the outcome of any one cross. If a single heterozygous flower is selfed, it may produce only 1 or 2 seeds. If you plant just one seed, there is 1 chance in 4 that it will have red flowers, 2 chances in 4 pink, and 1 chance in 4 white. The 1:2:1 ratio was one of the great discoveries of Gregor Mendel, an Austrian monk who performed experiments with pea plants. His crossbreeding experiments became the basis for modern genetics. Mendel discovered that in a selfing of this type, the recovery of the parental types means that genetic material must be composed of particles such that the R genetic material can be separated from the R genetic material in the RR heterozygote. Before Mendel's work, it was thought that the genetic material was a fluid. Two fluids cannot mix and then separate again perfectly. The constancy of the 1:2:1 ratio made it more logical to think in terms of discrete particles, genes, that never lost their identity regardless of the crosses in which they participated. Furthermore, the 1:2:1 ratio can be realistically interpreted only in terms of each individual plant having 2 copies, being diploid, and each sex cell having 1 copy, being haploid. If each plant had only 1 copy, pink-flowered heterozygotes would be impossible, whereas if each had 3, phenotypes such as dark pink and light pink should also be present. Keep in mind the state of scientific knowledge in 1865 when Mendel was working. The concept that all organisms are composed of cells with nuclei had only recently been proposed. Mitosis and cell division were very poorly understood, and meiosis would not be discovered for another 23 years. The concepts of chromosomes and homologous pairs would not be well established until the 20th century, and the existence of mRNA was not confirmed until the mid-1960s. Figure 16:11 A Punnett square makes it easy to analyze and understand the results of a cross. Gametes from one parent are listed across the top, those of the other parent are listed on the left side. Botany and beyond. Box 16:1 Botanical Philosophy and Popular Culture. When thinking about genetics and the contributions of Gregor Mendel, it is important to keep in mind the state of scientific knowledge at that time. Some of the greatest scientific geniuses of all time were discovering and documenting the fundamental concepts of biology. Only 27 years earlier than Mendel's work, M. J. Schleiden and T. Schwann had proposed the cell theory, the concept that all organisms are composed of cells with nuclei and that all existing cells come from pre-existing cells. We take this for granted today, but it means that all living organisms are fundamentally the same, that life may have arisen only once, and that all cells are descended from those first cells. In 1849, Wilhelm Hofmeister proved that during plant reproduction the new embryo develops primarily from the egg after receiving some influence from the pollen tube. This was revolutionary because until that time everyone firmly believed that in both plants and animals, the new embryo developed from the sperm cell, the female parent might provide nourishment and protection, but the new generation was really a continuation of the paternal parent. Hofmeister's conclusion that the female parent also contributes to the new offspring was both shocking and also very important for Mendel's later observation that genes are passed equally from both parents. In 1859, Charles Darwin published Origin of Species by Natural Selection, which postulated that organisms change through time, that they are not constant. The discovery of evolution by natural selection caused a great deal of trouble for Mendel. Natural selection requires diversity and change, whereas Mendel's theories of genetic inheritance showed that differences in phenotype were due only to the mixing and separating of unchanged, constant genes. The idea of evolution had aroused great controversy and serious philosophical battles between proponents of evolution and of biblical creation. This battle had been raging for 6 years when the paper by Mendel appeared. Had he published 10 years earlier, he might have found instant acceptance, and Darwin would have had a more difficult time. A further difficulty for genetics, however, was its postulation of genes as particles, not as fluids. This was at a time when fluids were considered the basis for most of biology, with sieve pores in the phloem and pits and perforations in the xylem considered to be effective by acting as filters that separated the various fluids that were thought to exist. Fortunately, in 1879, H. Fall showed that during angiosperm reproduction, a sperm nucleus entered the egg, and in 1888, E. Strasburger described meiosis, giving a firm foundation to the concept of genes being particulate. If genes are on the chromosomes, then suddenly it is possible to see how Mendel's ratios and independent assortment could occur. This was summarized in 1915 by T. H. Morgan in The Mechanism of Mendelian Heredity. Finally, in 1927 and 1928, H. J. Muller and L. J. Stadler showed that genes could be artificially changed by the use of X-rays. Thus, genes could be stable enough to permit Mendelian genetics to operate and yet variable enough to allow natural selection to operate as well. It is important to keep in mind that these were not the only biologists working at the time, nor were these the only discoveries. A huge volume of information was being generated, much of which concerned unusual, exceptional, or aberrant cases, and which thus made the fundamental discoveries more difficult to recognize as being important. Also, we must always try to appreciate the profound influence of the general philosophy of the time. A male-dominated society produced male-dominated theories of biology. It is likely that our own sincerely held beliefs are causing us to improperly evaluate some of our own biological observations. Monohybrid crosses with complete dominance. The situation in which only half as much product of an enzyme, such as the red pigment discussed above, is produced in a heterozygote is not universal. In certain species or with other traits, cytoplasmic control mechanisms may cause the enzyme to function until a specific amount of product is synthesized. The enzyme may have to work faster or longer, but the final amount of product is the same whether the plant has 2 functional alleles or only one. In other situations, the amount of enzyme might be monitored such that the non-mutant mRNA of the heterozygote is translated more frequently or rapidly. In either case, the phenotype of the heterozygote is like that of the parent with 2 effective alleles. That trait is said to be dominant over the other version of the trait, which is recessive. An example is height. A tall plant with a TT genotype produces sex cells that are all T. A short plant, genotype TT, produces only T sex cells. When the tall plant is crossed with the short one, all F progeny have the TT genotype, but they all have the phenotype of being tall, indistinguishable from their TT, homozygous dominant parent. We do not know what protein is produced by the T allele, but even with only one functional allele, enough product is made to permit normal growth. TT plants are tall, and the tall character completely dominates the dwarf character. Figure 16:12. In this monohybrid cross, the trait tall shows complete dominance over the trait dwarf. In a TT nucleus, the T allele may be transcribed twice as much as each T allele in a TT nucleus. The resulting mRNA may be translated twice as much, or the protein may work twice as long or twice as fast. A TT plant may contain a level of T protein far above the threshold for responsiveness. Most of the wheat plants in this field have blown over and are lying on the ground. This is a tall variety of wheat. Its stems are too tall and slender to be strong enough to hold themselves upright. The harvesting machine will have to cut the wheat so low to the ground that dirt and rocks may be brought into the machine, damaging it. Also, if it rains before harvest, the stems lying close to the soil may rot. All the wheat plants in this field are still upright and will be easy to harvest. None of these plants has lodged. This is a short variety of wheat. Plant geneticists obtained the seeds for this crop by crossing plants that all had short, strong stems. This field can be harvested easily, and there is little chance any of the wheat will rot, even if it rains before harvest time. Knowing the molecular biology of genetic systems, you can predict that when heterozygotes are selfed, 2 types of sperm cells and 2 types of eggs are produced. The Punnett square for the cross is as in figure 16:13, and a genotype ratio of 1TT, 2TT, 1TT is expected. The prediction is correct, but what will the phenotype ratio be? 1 out of 4 plants will be tall due to a TT genotype, and 2 out of 4 will be tall due to a TT genotype. Thus, 3/4 have the tall phenotype, and 1/4 have the short phenotype. Whenever a cross is made and a phenotype ratio of 3:1 is seen, we should suspect that the parents are heterozygous and the character shows complete dominance. Test crosses. Remember that we can only see an organism's phenotype. To discover its genotype, we must perform crosses, and the test cross is one of the most useful. When traits with incomplete dominance are studied, the genotype of any plant is easy to determine from its phenotype. If the trait has complete dominance, it is difficult to know what the genotype of any particular plant is unless the plant shows the recessive trait. Imagine that you are studying the inheritance of tallness. You have selfed some heterozygotes and planted the resulting seeds. In your greenhouse or garden, there are now hundreds of plants, approximately 75% of which are tall and 25% short. You know the short ones are TT, and if you need a plant with the TT genotype for experimentation, you know automatically to choose short plants. But if you need to experiment on plants with the TT genotype, how can you tell which they are? A tall plant picked at random is more likely to be a TT plant because there are twice as many of them as TT tall plants. Figure 16:13. In setting up the Punnett square for a selfing of TT plants, first establish the genotypes of the 2 parents. Then determine what types of gametes are produced and in what proportions, and fill in the squares with the genotypes. From the genotypes, the phenotypes in each square can be determined. The genotype can be revealed by a test cross, a cross involving the plant in question and one that is homozygous recessive for the trait being studied. All gametes produced by a homozygous recessive parent carry the recessive allele, which is unable to mask the homologous allele in the resulting F zygote. If the plant being tested is actually homozygous dominant, 100% of the progeny will be heterozygous and tall. If the tall plant is heterozygous, half its progeny in the test cross will be tall and half short. If the cross produced thousands of seeds, you might not want to plant all of the seeds, just some, and if a large enough number of seeds are grown, the chance of accidentally not choosing any of the TT seeds is very low, and if even one of the F plants is short, the test parent must have been TT. After the actual genotypes of plants are known, those that are homozygous dominant can be gathered and planted in special areas, kept free of all natural pollinators, and allowed to breed among only themselves. All of their progeny will be homozygous dominant and can be used in breeding experiments. The homozygous recessives can also be kept as a special line, being selfed and kept pure. Such groups are purebred lines and are both useful and valuable. It is not possible to maintain the heterozygotes like this because they do not breed true, that is, their progeny are not exactly like them. 1/4 are homozygous recessive, 1/4 are homozygous dominant and cannot be distinguished visually from the 1/2 that are heterozygotes. So far, we have considered only one trait, height, but for plants that are important crop or horticultural species, dozens or even hundreds of traits are catalogued. Seed companies maintain hundreds of different lines of corn, for instance, in which the genotype of many characters is known for each line. Many times, plants are collected from the wild, so nothing is known about their genotype unless it is immediately obvious from the phenotype. From looking at a few collected plants, however, it is not possible to tell which characters are dominant and which are recessive. Carefully controlled and recorded crosses must be made. When test crosses must be made on annual plants, the results are usually not known until after the plants have died. Their genotypes are then known, but the plants cannot be used for experimentation or breeding. In such cases, it usually is necessary to do both test crosses and experimental crosses simultaneously, not knowing which plants have the correct genotype for the experiment being performed. After the test cross results are complete, the parents that had the proper genotype can be identified, and the experimental crosses that involve them can be analyzed. Multiple alleles. Each gene may have many alleles, not just two, as in the examples discussed so far. A protein of average size consists of about 300 amino acids, so the coding portion of its mRNA must have about 300 codons, each containing 3 nucleotides. The gene is therefore at least 900 nucleotides long, not counting introns and promoters. At least 900 sites exist at which point mutations can occur, and of course, any mutation may involve several nucleotides. Consequently, the gene may exist in many forms, called multiple alleles. When genes are polymorphic, having multiple alleles, numbers, such as X, 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 and so on, are used rather than capital and lowercase letters. Certain mutations still result in the production of a protein with the normal sequence, but most lead to altered protein structure. Some of these proteins are similar to the original protein, perhaps having similar or even identical enzymatic activity. However, many carry out the proper reaction more slowly or are not accurately controlled by regulatory mechanisms in the cytoplasm. A normal, wild-type phenotype may not be produced. With multiple alleles, the concept of dominance is more complex. One allele may produce the proper quantities of the functional protein, whereas a different allele produces more, another produces less, and a fourth produces one with altered activity. Many different phenotypes are possible. Figure 16, 14 test crosses to determine whether a plant with the dominant phenotype is heterozygous or homozygous. Of the data in this diagram, you would not know the genotypes of the test parents before the test cross. Those data are what you are trying to discover. If you want to find only a single homozygote or heterozygote, you would need to do test crosses on only one or a few plants. Imagine, however, that you had done an experimental cross for a trait that you assumed would give you a 3:1 ratio. It would be necessary to do test crosses on a large number of progeny plants with the dominant phenotype just to confirm your experiment. Within a population of plants, as many types of gametes can be produced as there are different types of alleles. A heterozygous XX plant can be crossed with a heterozygous XX1, resulting in progeny such as in figure 16, 16, XX, 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 and XX. For distinct types of F plant are produced, none of which has the genotype of either parent. Figure 16, 15 maintaining purebred lines is relatively easy for perennial plants. After the plant is growing, it can produce pollen or ovules for experimentation for years. Annual plants are much more difficult because pollination, seed gathering, planting, and record keeping must be done annually. Here, wheat seeds are being frozen in liquid nitrogen. After several years, some will be thawed and planted. Their seeds will be frozen for further storage. Figure 16 with multiple alleles for a single character, numerous types of crosses become possible. However, we still determine, from the parental genotype, all possible gametes and then construct a Punnett square in the usual fashion. Dihybrid crosses. A dihybrid cross is one in which two genes are studied and analyzed simultaneously, rather than just one, as in a monohybrid cross. Every cross involves all the genes in the organism, but the terms monohybrid and dihybrid refer only to the number being analyzed. When two genes are studied, the results of the crosses depend on the positions of the genes on the chromosomes. If they are on different chromosomes, the alleles for one gene move independently of the alleles for the other gene, but when two genes are close together on the same chromosome, the alleles for one gene are chemically bound to the alleles for the other gene and move together. The situation in which the genes are on separate chromosomes is easier to understand and is explained first. Genes on separate chromosomes, independent assortment. Consider a plant heterozygous for two traits, for instance seed coat texture and color, with a smooth seed coat showing complete dominance over a wrinkled seed coat, and a yellow seed coat being dominant over a green seed coat. The plant's genotype is SSYY, and its phenotype is smooth yellow seeds. We know that if we consider only the gene for color, the plant produces two types of gametes in approximately equal numbers, some carrying Y and some carrying Y. Also, if we consider only texture, half carry S and half S. How do the alleles of the two genes relate to each other? As in monohybrid crosses, knowing the types of gametes that can be formed is the key to understanding the patterns of inheritance. If the two genes are on separate chromosomes, the alleles of one gene move independently of the alleles of the other gene. This is called independent assortment. The chromosomes have duplicated during S phase and each has two copies of each allele. All chromosomes align on the metaphase plate during metaphase I, where homologous chromosomes also pair with each other. There are two Y alleles, two Y alleles, two S alleles, and two S alleles at the metaphase plate. During anaphase I, homologous chromosomes separate from each other, and both Y alleles move to one pole because they are on the two chromatids of one chromosome, still held together by its centromere. Both Y alleles, located on the two chromatids of the homologous chromosome, move to the other pole. There is no way to predict which pole will receive which type of allele. Similarly, the S alleles separate from the two S alleles and move randomly to the poles. In some cells, the pole that receives the Y alleles at telophase I also receives the S alleles, but in other cells, the Y and S alleles end up together. During meiosis II, the two chromatids of each chromosome separate from each other, resulting in four types of haploid cells in equal numbers: SY, SY, Psi, and SY. Any single microspore or megaspore mother cell produces only two types of haploid cell, the set SY and SY or the set SY and Psi. All four types are produced by a single plant because some mother cells produce one set and some produce the other set. Figure 17 during anaphase I, chromosomes move independently of each other, so one pole receives S and the other receives S, likewise, one gets Y and the other Y. But in about half the cells, both S and Y move to the same spindle pole by chance, whereas in other dividing cells, one spindle pole receives S and Y, again by chance. A Punnett square for a dihybrid cross is set up just like one for a monohybrid cross. Establish the types and relative abundance of gametes, and then fill in the squares. The table looks a little formidable, but it really consists of 2:3:1 ratios intermingled. After the possible types of gametes are known, the Punnett square can be set up, as in figure 17b. Any single fertilization results from the syngamy of one sperm cell and one egg cell and produces only one of the 16 possible zygote genotypes shown in figure 17b. All 16 types of zygote occur only if we study many fertilizations. To have the ratios come out accurately, we have to analyze hundreds of progeny. In plants, it is usually easy to obtain large numbers of fertilizations because pollen and ovules are produced in large amounts. After pollinated, most plants produce dozens or even thousands of seeds, enough progeny that all 16 zygote genotypes occur in about the expected ratios. But in large animals such as mammals, reproduction may be infrequent, and only 1 or 2 offspring are produced each year. A great deal of work is necessary to get enough progeny to verify the results of a dihybrid cross. In a dihybrid cross involving independent assortment of 2 heterozygous genes, each gene showing complete dominance, a characteristic phenotype ratio occurs, just as is true of the 3:1 ratio in a monohybrid cross. The ratio is 9:3 to 3:1, with 9/16 of the plants having the dominant phenotype for both traits, 3/16 with the dominant phenotype of the first trait and the recessive phenotype of the second. 3/16 with the first trait recessive and the second dominant. And 1/16 in which the plants have the recessive phenotype of both traits, wrinkled. Green. The 9:3 to 3:1 ratio results only if all 4 types of gametes are produced in equal numbers and have equal opportunity to participate in reproduction. The alleles Y and Y must be independent of S and S during meiosis I. This automatically happens if they are on different chromosomes. Table 16, 3 genotype and phenotype ratios of a dihybrid cross. The 2 individuals are heterozygous for 2 characters showing complete dominance and independent assortment. The notation indicates that either allele may be present, example, SYY represents either SSYY or SSYY, both of which produce a smooth green phenotype. Notice that if only 1 trait is considered, it behaves as in a monohybrid cross. Plants with smooth seeds outnumber those with wrinkled seeds by 3:1, and those with yellow seeds are 3 times more abundant than those with green seeds. Similarly, the monohybrid genotype ratios are also present, 1SS, 2SS, 1SS and 1YY, 2YY, 1YY. Considering 2 genes simultaneously does not affect their inheritance at all. Crossing over. Independent assortment can also occur if 2 genes are located far apart on the same chromosome such that crossing over occurs between them during prophase I, after homologous chromosomes have paired and a synaptonemal complex is formed. Because no preferential sites for crossing over seem to exist, the farther apart 2 genes are, the greater the possibility that crossing over will occur between them. Most plant chromosomes are so long that crossing over occurs several times within each chromosome during each prophase I. Consequently, the 2 ends act like separate entities. If the gene for seed coat color were at one end of the chromosome and the gene for seed coat texture were at the other, they would still undergo independent assortment. If the 2 genes are close together on a chromosome, however, crossing over may not occur and the 2 alleles may move together during meiosis I, as described below. Genes on the same chromosome, linkage. If 2 genes occur close together on a chromosome, they usually do not undergo independent assortment during meiosis I. Instead, the 2 genes are said to be linked. Consider a plant heterozygous for the 2 traits of the seed coat again, but now imagine the genes occurring close together on one chromosome. During meiosis, haploid cells that are either S or S, Y or Y are formed, but a new complexity arises. Because the genes are linked, several types of heterozygote are possible. An SSYY individual may have the alleles S and Y on one chromosome and the alleles S and Y on the homologous chromosome, but an SSYY individual may have S and Y linked together and S and Y linked. We must consider the types of gametes that can be formed. If crossing over is ignored, the first individual produces haploid cells with the genotypes SY and SY only, whereas the second plant would produce SY and Psi gametes only. The result of selfing is not a 9:3 to 3:1 ratio, but something drastically different. Figure 16, 18 2 types of SSYY individuals are possible, but in most instances, their phenotypes are identical. Only genetic tests can distinguish which is which, based on the unique gametes produced by each. A test cross, with the plant on the left in being the test parent. Try setting up the Punnett square for a test cross with the plant on the right in. A test cross of 2 closely linked genes produces results very different from those of a test cross involving non-linked genes that assort independently. What would the Punnett square be like if S and Y were not linked? Botany and beyond. Box 16-2 Whose genes do you have? You received half your chromosomes from your father and the other half from your mother. So half of the DNA you had when you were a zygote, a single cell, was your father's and half was your mother's. But beyond that? 1/4 of your genes came from your paternal grandfather, 1/4 from your paternal grandmother, and so on for your maternal grandparents. But, can we say that 1/4 of your DNA came from each of your grandparents? That is not so easy. Of the thousands of cells in your father's testes, one in particular underwent meiosis and produced 4 sperm cells, one of which gave rise to you. During prophase I of that meiosis, the chromosomes your father got from his father paired with the ones he got from his mother, your grandmother, and they underwent crossing over. If each homologous chromosome were to break in exactly the same place, then when they are repaired and meiosis continued, each would have exactly the same amount of DNA. Often, however, homologous chromosomes do not break at quite the same spot. After repair, one might have a bit more DNA, the other a bit less, one might be mostly paternal, the other mostly maternal. As long as they are not too unbalanced, the chromosomes will be functional. But because of this, we cannot say that exactly 1/4 of your DNA came from each of your grandparents. Some might have given you a bit more, and others a bit less. With each generation we go back, the number of ancestors doubles, and the number of genes we received from each drops by half. By the time we get to the 15th generation, we have 32,768 great etc. grandparents, and each contributed 1 in 32,768 of our genes. We believe, however, humans only have somewhere between 30,000 and 35,000 genes. If we assume each generation is about 20 years, then this 15th generation lived about 300 years ago, about 1700 CE, not long after the first microscopes were invented. Of course, each of us is descended from many more generations before that, but even so, we may not have actually gotten any of their DNA. The most instructive cross is a test cross using a double homozygous recessive parent, SSYY. If we continue to exclude crossing over, the gametes from the first type of heterozygote will be SY, and the gametes from the homozygous recessive parent will be all SY. 2 types of F will be produced, as shown in Figure 16, 18b, smooth yellow and wrinkled green, and they occur in a ratio of 1:1. The 2 phenotypes are like those of the parents. If the genes are not extremely close together, crossing over may occur. For example, in 10% of the cells undergoing meiosis, a crossing over might happen. Therefore, the plant would produce 4 types of gametes, but not in equal numbers. Of the gametes for the first type of heterozygote, 47.5% would be SY, 47.5% SY, 2.5% SY, and 2.5% SY. The last 2 are recombinant chromosomes formed from a crossing over of the homologous chromosomes and recombination of alleles. The first 2 are parental-type chromosomes. In the majority of the cells, Y is still linked to S and Y is linked to S, but in a minority, Y has become linked to S and Y to S. A Punnett square alone is not sufficient to show the relative proportions of progeny, so the percentages of gamete must be added. A test cross now would result in 47.5% of the F progeny being smooth and yellow, 47.5% wrinkled and green, 2.5% wrinkled yellow, and 2.5% smooth green. The first 2 types are like the parents, but the last 2 are recombinant types produced at the same percentage as crossing over occurs. We cannot actually see crossing over with a microscope, but we can infer that because 5% of the FS have a recombinant phenotype, then 5% of the chromatids must have undergone crossing over. The rate of crossing over is directly proportional to the physical spacing between the genes on a chromosome. 2 genes that produce 6% recombinant FS are closer together than 2 genes that produce 10% recombinant FS. By analyzing as many mutant alleles as possible, we can measure space between them in recombination percentages, each 1% being called 1 map unit or 1 centimorgan. 1 map unit, on average, is approximately 1 million base pairs. By this means, a genetic map can be constructed. If enough genes can be mapped, we may find that A is linked to B, which is linked to C, even if A and C are so far apart that they assort independently. By knowing that both are linked to a common gene, B, we know that all 3 are on the same chromosome. On the other hand, if A, B, and C cannot be shown to be linked to another gene F, that does not mean that F is on a different chromosome. It might be that intervening genes have not yet been mapped. A set of genes known to be linked is called a linkage group. When all genes are mapped, there will be exactly as many linkage groups as there are chromosomes, but if only a few genes are mapped, there may be more linkage groups than chromosomes. Figure 16:19 with linkage, for types of haploid genotypes might be produced, but not in equal numbers. Recombinant types are less abundant than parental types, in this case 5% recombinant chromosomes and 95% parental type. These two genes are 5 map units apart. This test cross, unlike that of figure 16:18b, gives 4 types of progeny, 2 parental types and 2 recombinant types. The alleles S and Y show linkage rather than independent assortment. Figure 16:20a and C might be far enough apart for crossing over to occur in virtually all nuclei, so they undergo independent assortment. If each can be shown to be linked to B, they must be on the same chromosome. If A, B, and C are not known to be linked to F, it might be because they are on separate chromosomes or because they are on the same chromosome but intervening genes have not yet been mapped, as shown in. Because of incomplete mapping, ABC, JK, and STUV are 3 linkage groups even though there is only 1 chromosome. Multiple genes for one character, quantitative trait loci. Individual phenotypic traits are the result of complex metabolic processes involving numerous enzymes, therefore, many separate genes may affect any single trait. The gene R was described as affecting flower color by producing an enzyme that synthesized a red pigment, but that enzyme requires the proper substrate, which is present only if it is synthesized by a different enzyme controlled by a distinct gene. If this gene is present as an ineffective allele, there will be no substrate and thus no pigment, regardless of whether the first flower color gene is present as allele R or allele R. In Arabidopsis thaliana, abscisic acid causes developing seeds to become dormant at maturity. In several mutants, mature embryos continue to grow as if germinating and do not enter dormancy. One mutant, ABA, is unable to produce enough abscisic acid. A second mutant, ABI3, produces plenty of abscisic acid but does not respond to it, perhaps its receptor is the affected protein. Although ABA and ABI3 involve different genes, they both result in the same phenotype. They are multiple genes for one trait even though they are not part of one metabolic pathway. Table 16 for multiple genes for one character. Ing. Fresh weight of seed tissue. Most synthetic pathways involve at least 4 or 5 intermediates, and their 4 or 5 genes all affect the same trait. Complex traits such as leaf shape, cold hardiness, and general vigor can be the results of dozens of genes, and rather than having phenotypes that are simply dominant and recessive, the trait may show a continuous gradient of values. Having multiple genes for each trait is referred to as epistasis. Some intermediates may be produced by several pathways, each with its own enzymes. A mutation in one is not necessarily particularly severe because the alternate pathway may increase its activity and produce sufficient amounts of the intermediate. If this happens, the mutation is masked and is not reflected in the phenotype, even if it is present in the homozygous condition. Extremely complex crosses, involving hundreds or thousands of progeny, may have to be performed to determine what fraction of a particular phenotype is correlated with a particular gene. The genes or other portions of DNA associated with such traits are quantitative trait loci. Conversely, when an intermediate is part of several metabolic pathways and is produced by only one enzyme, a mutation in that enzyme's gene affects all of the pathways and alters several different traits. Multiple phenotype effects of one mutation are called pleiotropic effects. For example, any mutation that affects the protein portion of phytochrome affects all developmental processes controlled by phytochrome, and mutations that alter the level of pyruvate affect the citric acid cycle, amino acid synthesis, and C metabolism. Other aspects of inheritance. Maternal inheritance. In the crosses described above, the alleles of both parents are transmitted equally to the progeny, a situation called biparental inheritance. All genes in the nucleus undergo biparental inheritance. An unexpected feature of both plants and animals is that during fertilization, the sperm cell loses most of its cytoplasm and only the sperm nucleus enters the egg. Consequently, the zygote obtains all its plastid and mitochondrion genomes from the maternal parent. This is uniparental inheritance, more specifically maternal inheritance. Mitochondrion genetics are difficult to study for several reasons. First, each cell has many mitochondria, up to hundreds, and each mitochondrion has several circles of DNA. Each cell has multiple copies of each mitochondrion gene and may have received diverse types of alleles from its maternal parent. Simple Mendelian ratios are not produced. Second, the presence of many of the mitochondrion DNA mutants does not result in easily recognizable phenotypes. The plants usually simply grow somewhat more slowly, or if the mutation has a severe effect, they may die as embryos. Consequently, the presence of mitochondrion mutants may not be detectable by examination. Plastid genetics are somewhat easier if alleles affecting chlorophyll synthesis are studied. Consider a plastid whose DNA has mutated so that the plastid can no longer produce any chlorophyll. It may be red or orange because carotenoids are no longer masked by chlorophyll, or it may be white if no pigment at all is produced. If a zygote receives only plastids like these, the embryo and seedling will be red, orange, or white. Such seedlings are rare, but in large commercial nurseries that grow millions of seedlings, they appear from time to time. The plants can be kept alive artificially if they are grafted onto a normal plant that supplies them with carbohydrates. Grafted plants can then be used for genetic experiments. When an achlorophyllous plant is crossed with a green, chlorophyll-bearing plant, the outcome depends on which plant is the pollen parent and which is the ovule parent. If the achlorophyllous parent provides the pollen, 100% of the zygotes will be green because they receive all of their plastids from the normal parent by way of the egg. All mutant plastids are destroyed during sperm differentiation and syngamy. But if the achlorophyllous plant is the ovule parent, all progeny also have mutant plastids and are achlorophyllous. For some reason, conifers are an exception among all plants. Conifer eggs destroy their plastids, but conifer sperms do not. Therefore, in conifers, plastid genes are inherited paternally, mitochondrial genes are inherited maternally, and nuclear genes have biparental inheritance. Figure 16.21 Plastid mutations in the cactus genus Gymnocalycium seem to be common. Quite often totally red or orange seedlings appear in cactus nurseries. These lack chlorophyll but can be kept alive by grafting them onto a green cactus that supplies them with sugar. Many plants like coleus and maples have bright red leaves. They do have chlorophyll, but it is masked by large amounts of other pigments. Plastid inheritance is also responsible for certain types of variegation in plants, the presence of spots or sectors that are white, red, or orange on a plant that is otherwise green. Because plant cells have many plastids, only rarely do all the plastids of an egg have the same alleles unless the parent has only one type, as in the cactus of Figure 16.21. More often, each cell has a mixture of plastid types. During cell division, some plastids move into each end of the dividing cell. Usually random chance alone results in each daughter cell getting some of both types of plastid. However, occasionally, one daughter cell by chance receives only mutant plastids. It is white, red, or orange and is able to survive because surrounding normal cells supply it with sugar. If this occurs in a leaf primordium or a young internode, the cell continues to grow and divide along with the surrounding cells, resulting in a patch of cells that lack chlorophyll. If it occurs in a very young leaf, the cells grow quite a lot and produce a large spot. If it happens in an older leaf that has reached almost full size, the patch is small. When variegation affects stems, the achlorophyllous patch may include an axillary bud. If so, the entire bud and the branch that grows from it are achlorophyllous. Any flowers it produces can be used for genetics experiments, providing either pollen or ovules. Experiments on both corn and morning glory have shown the achlorophyllous character to be maternally inherited, thus, it is a feature of the plastid DNA as just described. Recombinant DNA technology is now greatly accelerating our efforts to understand plastid and mitochondrion genetics. In both organelles, the DNA is a circular double helix, one of its ends being attached to the other, and no histones are present. Plastids contain many circles, 200 in each spinach plastid and 1,000 in each of those of wheat, so rather than being haploid or diploid, wheat plastids are 1,000 ploid. Plastid DNA may constitute up to 21% of the total DNA content of a cell. In angiosperms, plastid DNA tends to be about 150 kbp long. In plants, mitochondrial DNA circles are very large, well over 200 kbp, more than 10 times larger than mitochondrial DNA in animals. Genomes of plastids contain genes for numerous enzymes as well as a complete set of plastid ribosomal RNAs and 30 of the transfer RNAs. Currently, it is estimated that 80 to 100 enzymes in membrane proteins are coded by plastid DNA, and many of these are essential, the large subunit of RuBP carboxylase, components of the proton channel ADP synthetase complex, cytochromes, and proteins that bind chlorophyll A. Mitochondrial DNA also contains genes for rRNA and tRNA, as well as those that code for proteins involved in ADP synthetase complexes, electron transport, and an ADH dehydrogenase complex. In many cases, organelle genes must